So for you guys now, there is an advanced JIPipe folder, and we will need this one. Ruben mentioned yesterday that it's possible to share templates. And what I did was I made a collection of all my templates and uploaded them for you as a present. Please download that file. It's just a JSON file, and we will set it up together how to put it into JIPipe, because on this JIPipe we don't have it either. And then we have two folders, neutrophils and cryogel. We will need hopefully both, depending on how the time goes. Probably the neutrophils. And then what we will do is, actually I have to download this one also myself because I forgot to put it on my hard drive. Okay, good. So you can follow me, and the code should live here. Cell tracking 01. This is the actual JIPipe code. Just open that. There is nothing open, so you can put it in this window, whatever you prefer. And then we will set up the user directories together. Yeah. So first of all, you will have an error message that your user directory is invalid. That's because this was set up for my laptop. As Ruben mentioned yesterday, it is possible to have project-wide folders. And he mentioned how to define them. So I will show you that in a second. I'm getting a complaint here. Yes, it's my project. Yes, whatever. Okay. So if you remember, these settings are in the project overview tab. And here, what we will need is to set up these directories. So if you don't see it, this is what we need. So you go there, and then you go to configure. And I did set it up yesterday. So you have 3 user directories. One is called analysis folder. One is called data folder. This basically means that from the user's point of view, I like to predefine where my data are going to be and where my analysis is going to go to. This has the advantage you will see in a few minutes that if you get new data set, or if you decide to reanalyze your data and now you go to a different analysis folder, you do not have to change these folders throughout your project. You only change them once, namely here, and then I will show you the node with which to read these into the workflow, and then you are done. Now, on the other hand, if you have a complex workflow, which you will see today, where you need this information more than once, then this would mean that somewhere you will for sure either forget it to set to the new folder, and then let's say the analysis goes to the wrong folder, or the program just gives you an error message that this folder is not there. So this is, for me, easier to set it up at the very beginning. And the way you set this up is very simple. You give the folder a name that's logical to you. I call them data folder and analysis. Call them whatever you want. But the important thing is that then you have to find where these folders are. And obviously they are not on the C drive. This is from my laptop. So basically you have to navigate to the folder wherever you downloaded the project that I gave you. And if you navigate to the project I gave you, there is a neutrophils subfolder and there is an analysis subfolder. I want this one to be my analysis folder. So I select it and I say open it. And now my analysis folder, custom folder is set up. Yeah. And then I do the same for my data folder. I go back here, I go to data, and that's it. We are set. We say, okay, open this one. And now we updated both our analysis folder and our data folder. So from now on, throughout the whole project, throughout the processing that you will do in the next few weeks, months, whatever, this is how you will refer to these folders. And if you change them here. It takes you 5 seconds, 10 seconds, and you're done. Okay, so now we have this one. We close it. And from now on, it should be happy with us. And then let me briefly talk to you about the compartments. If you remember, to organize complex code is much easier if you put the code into compartments. to me, it was interesting that when Rumen introduced the concept of compartments, I was like, you don't need them. Why do you need them? And then Rumen was, you will see, when you have a complex project where the connections look like, as he always says, vomited spaghetti, then you will say, okay, I need to organize this somehow better. And from that point on, we always followed this process where we separate the logical steps of image analysis. In this particular case, what we will be looking at is neutrophils, which microbiologists know this much better than we do, part of the immune system migrating in a petri dish. It's a very simple experiment, but it's a good example to teach JIPipe. This is actually a real project which we published where we studied the effect of certain changes in the membrane structure that affect the motility of the neutrophils, under the effect of which sometimes they move faster, sometimes they move more slowly. So basically, the analysis I divided into these 5 compartments, and then we will go through them, and you are very, very welcome to follow through. So first of all, we go to the pre-processing compartment, and if you double-click on it, it will open a tab, and now this is how the project then begins with the pre-processing. So first of all, what you have not heard about probably yet is how to read in these user directories. There is a node which is called project user directory. If you run this one, then what you will find is that it identified the folder that we just updated to. So JSC, Zoltán, neutrophils data. So it knows now where we are. And what this node does is simply grabs this custom design folder and puts it out through the output. And from now on, I can then start reading in the data. Now, what you will see here is that instead of this array of 5 or 6 nodes that Rumen showed you yesterday, where you read the folders, the subfolders, the images, and then you filter them based on name, for example, you will see one node. And this one node is a group node. It's a group node that I generated. Before we go to that, I also wanted to show you how to get templates shared by somebody. So basically, if you go to the templates tab, this one here, then there is a way to get files in there, namely import from file. The file I just gave you, this one, JIPipe global templates JSON, what you then do is open it. Then it will ask you, do you want to use these templates just for this particular project or through the rest of your life? And then in the latter case, which I would recommend, just choose globally. This means that all the projects will have access to it in this instance of JIPipe. If you open or install a new JIPipe, this has to be repeated. But this has the big advantage, this whole system, that as you work on JIPipe longer and longer and you create your own templates, you can always carry them on with you to the next version of JIPipe. You can also keep a copy of it, which is what I do online in your drive, so that you have access to them everywhere, because a lot of these tools will take you a lot of time to develop, and you don't want to develop them again and lose them. So what happened here now is that suddenly in the templates tab, instead of having 2 sad little files, now we have a whole bunch of them. We will not go through them, but this is for you, free to use as part of the course. These are the templates which I developed over the past couple of years. Many of them will have 0 use to you. I did not go through them to select, because I don't know what you will need later. But you are free to go through them, and then the names are typically telly enough so that it will tell you what exactly they do. And one example is this read group folder. And if you double-click on this here, it will open what's inside, and you saw this yesterday from Rumen. These are the steps that we always use when we load data. Namely, we take the folder from the input, and then, as you already saw yesterday, we remember where the data came from. We put an annotation for the folder. Then we list the subfolders, And we also remember the name of the subfolders. Annotation subfolder. Here I have an extra node where I also remember the full path. So this will be like d/JSC/Zoltán, blah, blah, blah. So the whole thing, just in case you need it later. And when we explore the structure, then we list the files. And this you saw yesterday. But as you see here, I did not put a filtering expression because I wanted this to be intact. And then instead what I did was, once I remembered the image names as an annotation, again, this is with a hashtag, so it's an important annotation, because usually you will collect your data per image. I put an extra node called filter paths. And what this one does is it allows you to put in all kinds of filtering strings that will determine that from the folders, subfolders, and images, which ones do you want to work with. And this means that you can have your entire folder structure put into and let JIPipe know this is what I'm going to work with. But at this moment, I don't want to work with all of them. I will set up the processing for the weekend or overnight. I want to work with one or two, set up my workflow, check if everything works, and then move on. But for that, you need a way to tell JIPipe what do I want to do. And this was already explained to you, so I will just show you what I have done here. So what I did was simply that. First of all, I said, maybe there are a bunch of images in the folder somewhere. You didn't clean them up or you whatever. So you need a filter, and you saw this yesterday, which checks the image type. So you say, okay, this is the extension of the image. This must be in the image annotation. And since the annotation was created above here, the program knows already what image is. And it will check, does it end with CZI? And then I wanted to select a subfolder. This is, for example, a replicate or a patient who gave the neutrophil examples. And you want to test that. So now you limit your analysis to this particular subfolder. And we know what subfolders are because we made an annotation earlier. And finally, I limited this to one image because we don't want to sit here all day with that. But when you are ready to analyze the whole subfolder, for example, then this part would go. And all you would do is go to this node and delete it. You would keep the rest of it. Or if you want to analyze the whole folder, because you set it up for overnight, you would also delete this part. Then this would go. I would keep this still because you never know. Very, very logical. The commands you will learn very quickly, but also there is an edit button here. You saw this yesterday, which tells you all the functions. And use my notes and templates to learn these also. The point is that these are the nodes that live inside my group. And what they do is, I can close this one now. What they do is when I run them, they will give me the target of this specific filter. In this case, it found one image called experiment 296 CZI. That's exactly what I wanted. It ignored everything else. Now I set up a second one here. And if we go inside that, then you will see that this one has no filtering at all. So if you use it this way, it will read in everything and everybody. So when we close that, I only uploaded one image, but what you will see is that it found a different image. And why does it do that? Because the filter here is now different. What I did was I said, now I look for TIFF images. I want to use the same subfolder, and I'm looking for one particular file, namely a projected image. You will have the option to either run this group folder, which will read the CZI file, or you can run this group node, which will load the TIFF file. And TIFF file is much smaller. So we are going for the rest of the presentation, we are going to follow the right path, not the left path. But I leave the left path there because I want to show you how to make files smaller or project them or select certain parts of let's say, a time series during demonstration, you may or may not be able to run the left side of it, depending on whether you have the CZI file and whether your RAM is big enough, but you should be able to follow the right side. So what do we do here? We do a filtering, and as a result of that, we have a file which is our projected TIFF file. As the name says, this is 120 time points, but it's not a Z stack anymore, but rather it is projected into 2D. And from now on, I don't have to keep copying those 7 or 8 nodes all the time, but rather just put this one group node. And this you also find in my collection of templates. If you do a search for read, then it will find all those versions which I wrote over the years. They differ in terms of what they put out. Some of them will put out just the file that it identified based on the filter. The other will put out the files as well as the subfolders, so you can have information what the subfolders are. Just study them when you would like. But for example, this one will do the exact same thing, but it will also output the subfolder names in case you want to use it. So now we have found the file that we want to work with for the rest of this demonstration, so we can import it. Ruben showed you the import image node. I typically prefer the bioformats imported. That's the same as in ImageJ. The options are as usual. You can determine the order at which you load the images. These are basically Z stacks over time. So I set this one to XYZT. It doesn't matter terribly much. JIPipe is smart enough to figure this out, but I did set it up to be proper. And when you run it either way, because the previous node is already loaded. So typically during debugging, I use the cache intermediate results so that I see what happened beforehand in case there's a problem. In this particular case, it won't matter terribly much. And this will open the image. And if you double click on it, you will see that these are neutrophils. You can set the contrast as you would with any visual program. And then if you run the little black triangle here, then you will see the neutrophils moving around, being happy. This is, I think, one of the control examples where they were not limited in their mobility. But then you will see them moving, and the idea would be we want to find them, and we want to track them in time. And from the tracking, I want to know the speed at which they move, how far they are from each other, how zigzaggy their paths are. Do they move straight? Do they wander about? Do they move randomly around? All kinds of things. And this is the target of this project. And like I said, this is a real project that we published, I think, last year with collaborators. So now we have the image. And again, the reason I did this projection for you guys last night is that the CCI file is much, much bigger. So these steps would take 10, 15 times longer to execute, and that's not the goal. But when you have time, feel free to run the left side. It's exactly the same as the right side so far, and experiment with it when you like and when you want. Now, the next step is I want to know about the images, how big they are, how many time points there are, and so on and so forth. And this, by default, we don't know yet. So there is a special node, annotate with image properties. This is almost always in your workflow because you want to know a lot of things about the images. So if you look inside that, so you highlight it and you go to parameters, then what you will see is a whole bunch of options that you can learn about the image, 8-bit, 16-bit, etc., the color space that Ruben talked about, how tall and wide the image is, how many Z layers there are, and so on and so forth. But I only had interest in one parameter, namely how many time points there are. So you can, if you want, click all of these on, and then you will have all of this information saved as an annotation. How do we know it's an annotation? We know this from this sign here. So when I said, okay, I need to know how many time points there are, and I enabled this one, it is called by default image frame count. You can rename this one. This is accessible for you. Or you can just accept it as the default name. It's probably less confusing later. And what happens is that once I did that, and I run this node, what I will have is, on the right side in the cache, I will have, it's the same image. We didn't do anything with the image, but what I have is a new column called image frame count. It's an annotation. It's just like Ruben explained this morning. It's in the annotation table. And it says there are 121 time points. So now I can refer to this parameter later in my analysis, for example, to cut a certain time range from the data. And now I want to do exactly that. Let's assume you set up your example and you monitored your neutrophils in a tissue, for example, or organ on chip, or even in a petri dish overnight. You monitored what they do over many hours, and you have hundreds and hundreds of time points. You are setting up your workflow. You don't want to always do the calculations for all hundreds of images. So what you can do is you can limit how many time points you are using at any certain time. Why we can do that? We can do that because we know how many time points there are in the actual data set. So we put in a node which is called split by dimension. What we have so far is, originally we had a Z-stack and we had a time series. And I can say, okay, please cut this time series into individual images, each image corresponding to one time point. And for that, there is a node called that, split by dimension, and it does exactly as the name says. If you go to the parameters, it says split by time. We have other options, of course. You can split by channel, you can split by Z. You could also use this one, for example, if you have a very wide Z stack and you want to avoid the beginning and the top of the Z stack, you can say split it by Z and only keep those where the Z is between 10 and 30 or whatever. But what I'm doing is I'm splitting it by time. And then when I do that, what I will end up with is not one image, but 121 images. These are one image per time point. And then I say, I don't want to keep all of them. I want to keep some of them. And if you remember, the time dimension was called frame. We named this here when we split by the dimension. So now we have an annotation called frame, and I can carry this on to here where I says, I want to filter by annotation. There are other ways to do this using hyperstack cutting, but this actually works quite easily and easy to understand. And basically what we did was we put in this annotation. Namely, I said, for today's lecture, I would like you guys to see what happens between time point 40 and time point, the total number of frames minus 60. I knew it was 120, so this is going to be 60. If you don't want this filtering, then what you can simply do is go here, what I did on the left side. You can go here and you can say, I don't want to filter anymore. You can, of course, just delete what's in this box. Or you can go here and you can say, okay, I want to pass through this node. This means the data will flow through it. Not all nodes allow you to do that, but those that do, it's very convenient because you don't have to delete it. You can test, for example, extra steps in your analysis, and some of them you just turn off, go through it without having to disconnect it, move it away, reconnect the remaining nodes. This is a major pain in the neck. You simply pass through it. And if you pass through it, then instead of having 20 images, now you have all 121 of them. But let's make this faster, so we turn this back on. Okay, so now we have 20 images out between time point 41 and 59. Now, these are transmitted light images. The microscope was set up, it was aligned, but because of illumination inhomogeneity, this corner is a bit darker than that corner. This will mess with your segmentation layer. We will use a Cellpose-based, deep learning-based segmentation tool, but either way, whatever you use, they don't like it when you have an illumination gradient or an illumination inhomogeneity in the image. So with transmitted light images especially, it's very, very common that you correct for the illumination inhomogeneity. So what we'll do is we will use the node that our friend wrote specifically for this one, illumination correction 2D. And if you look at that, there is not a heck of a whole lot to set up. But one important thing is that to determine to which level I want to correct for illumination, we have to tell the program what is the radius within which I look for inhomogeneous illumination. And what we don't want is to remove the details of the transmitted light image. I just want to remove this large-scale inhomogeneous illumination. So I set these filters to quite large. If you set them to very small, they start to remove the actual contrast details of the transmitted light image. So you set these to 20 or 50, whatever then fits. But then what comes out at the end of this one is a set of images where this inhomogeneous illumination is not there anymore. So the left side is now the same brightness as the right side. And the segmentation will like that. And now I'm done pre-processing my images. And as you see, we are getting to the end of the compartment. We are getting to the output. There's one more thing to do. This is a time series. I need these guys after pre-processing to go back into a time series. I cut them up into individual ones, you remember. So I put in a node which will do exactly as the name says. It will take these two-dimensional slices and puts them back into a hyperstack. Hyperstack, you know, the same concept as ImageJ has. So what I will do is I will look at what I wrote in the parameter set. What I want to do is put the time frames into a hyperstack. A hyperstack is an image which can have channels, Z layers, and time points. There are no channels. There is only one channel. So I have to tell JIPipe, leave the channels at 0, leave the Z at 0 because this is not a Z stack, but do set up a time series based on the annotation called frame. And this frame, you remember, goes from 1 to 121 or 40 to 60. But the program knows that these are the time points. So it will use this parameter here to generate a stack. And if I run this one, then what I have at the end is a time series. A time series of 20. So this one runs from 1 to 20. You remember we had a filter earlier, which we turned back on. If I turn that filter off, then this will be 121, but then it takes longer, like I said, so it doesn't matter. But now we have the images. As you see, and I would like to emphasize that again, is that this is transmitted light image. It costs us exactly 0 to record them if you have fluorescence in addition to that. But with a properly aligned microscope, you will see a tremendous amount of details about these cells. And this is nothing special. This is just a confocal microscope, which in transmitted light mode is not confocal. But what it does is it really resolves the cells very well. So this is very important to remember also in microbiology, that it does pay off very often to record transmitted light images in addition to fluorescence. For example, it will tell you where the cells are, which fluorescence may or may not. So we have our friend that we want to work with, and this is the end of pre-processing. So what I do is I will link this output into my output node. And as you notice, this I wanted to emphasize, I set up my output node in a way that it inherits the original data. It has a slot called raw, which comes simply from the raw data from here. It has an input called Z projection, and this is not connected. And it has a connection called pre-projected, and this is where I connected my data. I called it pre-projected because I did exactly that last night. I pre-projected data. I want to have a pre-processing compartment which does all this stuff, so I don't have to worry about it anymore. You will use this very often because you will see that some tools in ImageJ and then correspondingly in JIPipe will not work, for example, on time series. Some plugins don't understand that it's a time series. Then you have to apply this operation or plugin on individual images. Or it doesn't understand the Z stack. And then you have to cut your stack up into individual layers or individual time points. So this is a tool that you will use often to allow certain processing to be done, which cannot be done on a complex hyperstack. Okay, so we have our output. Now, one more thing I wanted to note you that is that when I was setting this up for you last night, I realized, like I said, this is going to take too long, but I didn't want to delete this input to my output node. But I also didn't want to have to go through this. I can go through this and explain you, but at this point, it doesn't matter. So what I did was I disconnected the output of the left side processing, which would normally connect here. But JIPipe won't like that. So what I had to do was I had to go to this slot and I had to make it optional. So basically when you go here and you go edit this slot, then you have an option to make it optional. This means that you either connect something to it or you don't. It will not yell at you. It will not hate you for it. But you can connect. And then in the next compartment where this output will go to, now you have a choice. Am I going through the simple one for teaching or am I going through the complex one? And then I reconnect this one here. But it will not make me have to make a new output, for example, which only has two inputs too. So I can make this very flexible. So when we run this one, it will generate the projection, it will generate whatever it has to generate, and it will put it out into an output node. And as you remember from the lecture earlier from Ruman, compartments behave as they do in terms of nodes. Their output slot can be connected to one or multiple further compartments. So what is the next logical step? Now I want to find my neutrophils. We will be looking mostly for the round ones, but regardless, and I want to track them. So what do? I make a new node instead of piling stuff into the first compartment. I call it tracking, and I build it up. And we will go through this without terribly much details about the tracking itself. It's just using an ImageJ plugin. But basically, what we do is we grab the output from the previous compartment. And this appears, as you saw yesterday, as a node. It has three outputs. The Z projection was made optional, so it's not complaining that nothing is coming out, and I'm not using the Z projection anyway. But this has the advantage that my tracking compartment can be used without touching anything in it for the more complex analysis by simply connecting the Z projection output to the first node and disconnecting this one and then running it. All I would have to do is connect the other output, the left side pre-processing to the output node, and then I could run the exact same tracking compartment without changing anything else. But we are going with the pre-projected one because that's easier. And now you know what we are doing here. We are cutting them back again into individual time steps. Why are we doing that? You will see in a second. But the content of this one is the same that you saw before. If we go here, then you will see that what I do again is to cut by time. So now we end up with 20 individual images again. We run it. Now we have 20 images, as you see here, 20 rows. And then again, I added the opportunity to filter them further in case I didn't filter how many time series steps I do in the pre-processing. I just copied that node here, but as you see, if you look into the node itself, you will see that we are not filtering at all, because this is the maximum number of images. This, of course, when you do it more elegantly, will be replaced by the annotation image frame number, not 121. At the end of this one, we just end up with the same 20 images. We didn't do anything. Actually, I filtered out the first one, apparently. Why did we do that? We did that because I would like to use a segmentation tool called Cellpose. Cellpose is a tool that was developed originally as a standalone application, but now it's also available in ImageJ. It's a deep learning-based framework that was trained on literally millions and millions of biological images to recognize cells, nuclei, cell compartments, etc. Now, there are a lot of types of cells, so they train them on a lot of types of cells. That included neutrophils, macrophages, whatever. This was done over many years. The images were hand annotated, so people drew around, okay, this is a macrophage, this is a neutrophil, and they fed this into a deep learning system, and as a result was Cellpose. Cellpose requires two things. Cellpose requires the images that you want to segment. This could be fluorescence, this could be transmitted light, and Cellpose requires a model. What is a model? A model is the output of a deep learning system that was trained on a certain configuration or certain type of biological images. For example, cytoplasmic signal, or nuclei, or tissue, and so on and so forth. To use this as a standalone is possible. You can also program it in Python, but it's much, much, much easier to use it in JIPipe. So even those who do not use JIPipe so often, for Cellpose segmentation, they still go to JIPipe. And how do we use Cellpose segmentation? This will be so useful for you later. There is a node called Cellpose segmentation, and there is a node pre-trained Cellpose model. Now, how do we find these guys? We go back to add nodes, and we do a search for Cellpose. This will give you a list of nodes that are available for Cellpose users. There is a version 3 and there is a version 2. Right now I'm using version 2. I have not tested version 3 yet, and 2 works very well. And as you see, there will be various options. Let me focus on version 2 for now. There is a node for segmentation training. There is a node for segmentation. That's what we are using. There is a node to import models from files. And there is a node to access these pre-trained models that came with the system that the scientists trained on millions of images, or millions of cells. We don't go into details of all of them, but what I need right now is a node with which I can segment my transmitted light images without having to train the network. I have a handful of images. In this case, I have 120. Even if I wanted to train a deep learning network, I don't have the data for that. Plus, these are not particularly difficult images. These are neutrophils. So I assume that Cellpose will know what a neutrophil is. So I say, I'm going to go directly to segmentation. And like I said, it needs 2 inputs. It needs the images that I want to work with, and it needs a model which was pre-trained. Now, in terms of the first one, when this was written, it did not understand time series. So what you had to do is what I did here. Split the time series into individual images. Do the segmentation, and then later, you see here, now you are familiar with it, we put them back into a time series. Now, what does this guy have inside of it? What do you have to set up here? It's not terribly much, actually. These guys, 3D segmentation, enable multi-channel, these you don't need because I just left them on, it doesn't matter. But what you need is to give Cellpose a guidance how large your objects are. Because otherwise, if you have a mix of objects, let's say you have fungal spores and you have macrophages or neutrophils, there is an order of magnitude difference in the size. And you can help Cellpose find these objects by telling them how big they are approximately. You don't have to be very precise. So 30 means it could be between 20 and 100. But if you don't give it any guidance, then it will find everything, and then you have to filter it later yourself. It is possible to analyze two-channel images where you label the nuclei and then, let's say, the cytoplasm. We do not have that, so we don't need to set this up. And importantly, you have to decide what the node should output as a result. And these are the possibilities. So if you say, I simply want the segmented data to be output as a region of interest, so the outline, like you learned yesterday, then you click this on. If you want to learn more about how Cellpose works, how well does it recognize the cells, for example, then you can turn other outputs on, which, for example, will show you a probability map where the bright pixels will belong to the cells that it identifies, and the dark ones will say these are not neutrophils. This we will not use. I leave it on to show you later, but to test and fine-tune Cellpose, these are important, but for now we're not going to go into that. But what we need is the outline of the cells that it finds. Once we have that, we also have to tell Cellpose which model to use. And if you click on this node and then you choose your model, if you go here, you will see that there is an array of pre-trained models that you can choose from. Now, how do I know that Cytoplasm 2 works the best? Per default, you don't. You test them. And how do you test them? Well, you have a 120 image time series. You select one image out of it. You remember there is a filter on top of this compartment where I can further limit my analysis. I set this to 1, let's say from 40 to 41, whatever, so that the program runs fast. And then I test these various models. And this is how I back then decided Cytosol 2 is the best one to find these. Now, once you decided that, then you can say, okay, I'm done with testing, and I can set this one to my favorite model, which was Cytoplasm 2. Once you have that, you can simply say, this one is now set to the optimal one, Cytoplasm 2, and then we go back here, and then I can run this one. This one is done. So now you have the output images. And what comes out of this one is, as we expected, the regions of interest for those neutrophils that the program has found. As you will notice, these are all fairly round. And as I mentioned, Cytoplasm 2, this model that I used, mostly recognizes the round neutrophils. If you remember when we played the original video, some of the neutrophils will then flatten out as they are hunting for pathogens. Those we are not picking up at the moment. But then basically we have the identified regions of interest. We also, if you scroll down here, we also have the probabilities. In the cache, when you run a multiple output node, if you click on this tab where it says all outputs, it offers you the option to only show one of the slots. So if you click on all, then the cache will have all of them shown, but I can also just choose ROI, and then it only shows me the region of interest, and there's 19 of them. Now, what I wanted to show you, so the ROI we saw, this does what it's supposed to do, finds the neutrophils. But if I, for example, look at the probabilities, then this shows you a map as an image. What this one does is it tells you that the brighter pixels have a higher probability to belong to a neutrophil. So for example, this area here is just background. These bright ones, the deep learning model is sure that it's a neutrophil. These ones, it's like, yeah, it's probably one. And then as an image analyst, what you can also do is if you're not happy with the region of interest that it finds, you can use this map to do your own segmentation. Because now it's simply intensity-based segmentation. So you would denoise it and run automated threshold on it. We sometimes do that. And the last output which I ask for is the so-called flow. which shows you these little rainbow-colored structures, each of which corresponds to a cell. Now, when such a disk has the full spectrum like this, to put it simply, it means that Cellpose knows what this is. It has seen it before. If you show it an image which it has never seen, you show it the image of a frog, it will not have this round, complete spectrum because it doesn't know what it is. And this is excellent to judge which model you want to choose when you run it on multiple models. I didn't look at the ROIs when I tested it with the 15 models. I looked at the flows. Does this pre-trained model know what this is? Has it seen it? And Cytoplasm 2 was the one where I got these rainbows most complete. And as you see, it's really doing a good job. Even the ones which were dimmer on the probability map, it still has the proper rainbow range. What this means, you can again Google it or ChatGPT it about Cellpose, but this works very well. And based on this, we can then find the neutrophils. And that's why I always have these three outputs, not just the ROI, so I can see what's going on there. Now we know where the cells are, and now I want to track them. And to track them, I need not the ROIs, but I need masks. I need objects, binary objects in an image so that the tracker can track them. So basically what I do is I convert my regions of interest into a mask, basically, a black and white image. And now we have to remember, we are going to track them, but for tracking we need a time series, and we are not at a time series. We are individual images again, because Cellpose needed that. So we have to apply our old friend here, put them back into a time series. It's nothing new here for you anymore. It's just setting up the frame and leaving the channels and the Z at 0. So at the end of this, we will have our time series back. As I'm moving around, some of them time to time you miss, so the segmentation or the tracking will not be 100% perfect, but this is then where you later play with the parameters. And the rest of this here is basically nothing else but an implementation of TrackMate 7. Basically what it needs is a mask which corresponds to the objects. That's why we made a mask out of the ROIs. And it needs an image on which to do the detection. So basically what we do is we take our binarized hyperstack time series image, and we have to instruct the tracker what we are looking for. And we say we are looking to identify spots which we can track in time, and these spots should come based on a mask. So what I say is I need to detect masks, and then I need the image on which to detect them. And when you run this one, what it will do is it will identify the neutrophils based on the binary image, the threshold image, as objects which can be tracked over time. So if I run this one, these are the identified and tracked neutrophils. The ones which are flashing, they are there for too short, and then the program loses them. For example, they became very flat and Cellpose didn't see them anymore. But the stable ones are the ones that we will be able to then track. So what the tracker did was it identified these spots which can then be quantified. You can filter them. You can say, okay, I do not all of these spots. I want those spots which are above a certain size, for example. Now, I put this here for teaching purposes. The value is set to 1, so this is trivial. I want my detected object be more than 1 pixel. I mean, that's not a big requirement. But you can set this one to higher, and then you can exclude the very small objects. And once you did that, you can then simply track them. There are several models for tracking, just like there are several models for Cellpose. And you have to tell the tracking node, which is called track spots, which model to use. And I chose LAP because I went through the possibilities, and this was the best one. How do you know the possibilities? You do a search in your nodes, Kalman tracker, nearest neighbor, LAP, overlap tracker. The last one, for example, assumes that the spots overlap over time. Did I know which one is the best? No, I did not. I tested them all. So what I did was I went here, right-clicked on it, and I said find matching node. And then it will show you what you can connect there. So then the next step is we track them. It's very fast because we limit it to a few number of images. And now we have the result of the tracking. Nothing earth-shattering because we have very few images because we want to make it fast. But basically what you will see, the ID of the object, of the spot, the tracks in color, but it is very crowded. So you can go to spots in this tab here, and you can turn off the labels. You don't need the labels anyway. And now you have the individual cells outlined in red, and the colored line is the track that the center of the cell follows. Again, it's not very impressive because I don't analyze the whole time series, but feel free to do this on your own, and then you will see much more movement. But basically, the principle is the same. You now have the tracks that the cells follow throughout the observation of the images. And it was very, very straightforward up to now. You have various options here. You can also remove the spots with this one, or you can turn both of them back on. The same is true for the tracks. You can display them or remove them. With this one, you can also set the color of the tracks based on whatever, confinement ratio, and so on and so forth. Okay, so we have a nice tracked situation here. We can visualize them. There is a further option now to filter the tracks themselves. For example, what I did here was I wrote a dummy filter, which would say, okay, I need the track to be at least 2 time points, and it cannot be longer than the number of time frames. Or in this case, it's okay, make it sure it's at least as long as the number of time points minus 110. So this comes out to 10. This is up to you. You can say, okay, I only want to see those tracks which are almost as long as the video itself. So I don't track those neutrophils which we lose during segmentation, for example. But I turned this off because at this moment I just want you to see all the tracks that we found. And then there are 2 nodes which we don't really need. I turned these off. But basically then we are done. And we have our tracks saved, and we have the raw images saved. And I also put out the mask just in case we need it later. But now we are done with the tracking. Very clean output. Nothing that we don't need is saved, but everything that we need is saved. So if you go back to the compartments, now we have various options. We have the tracks. What do we want to do with them? For example, we can measure them. Speed, length, confinement ratio, zigzagginess, etc. Or we can visualize them. Make nice videos, put this in your supplement, in your paper, or your group presentation. Or we can process them further. But what you will notice is the principle I follow. I put all these different possible further processing into separate compartments. I could have done the measurements into the tracking and continue, but then the nice cleanliness is gone. So let's see what we can measure. So we go to the measurement compartment, and this is what it looks like. From the tracks, you have 3 measurements. You can measure the edges. An edge is the line between 2 tracking points, so 2 time points. The track is itself the whole array of edges that the cell follows during the video. And the spots are basically the identified binarized cells themselves. You can measure their properties. We will focus on measuring the track properties. So these 2 I disconnected last night, and they are just saved out. but we will focus on measuring the tracks. Now, if I run this one, this will just grab the results from the output. So the running it took literally a second. And now we have the output from the measurements. And this is what you get. A lot of data, whatever TrackMate 7 saves. But what you will notice is that there is a track ID and there is various parameters. For example, track displacement. How far does the cell go from time point 0 and time point end? In this case, it's only 20, so they don't go very far. The farthest one went 25 pixels. The shortest one didn't really go anywhere. It just wobbled in one place. So all these parameters then can be measured. You also have, for example, the speed. This is the fastest one. Then there's the maximum speed, and so on and so forth. These are measurements which come out of this one, and I can then, on the one hand, save them, and on the other hand, I can plot them. And just as an example, I put a histogram plot here, which will show you the median speed of the neutrophils. Okay, so here we see the median speed, and on the vertical axis, we see the frequency of those neutrophils which move at this speed. It's a histogram, yeah. And what you see is that most of them don't move terribly much. Then there is a group of neutrophils which move around 2.5 microns per minute. And there are a few neutrophils which are very fast. You can set a lot of parameters for this. You can, for example, increase the number of bins, then it's easier to identify the groups. You can set what is plotted. You can set labels, for example, what to print on the vertical axis, what to plot on the x-axis. Yeah, so for example, the x-axis will be called median speed, and the y-axis will be called frequency. But this is up to you. But the point is that you can programmatically set this one by this node. And this node allows you to, for example, generate a name for the data set that you are plotting. But I want this name for the data set to be specific to the data set that I'm using. So in this case, I wanted to know which model I was using for the tracking. So I used the annotation source slot, which I will show you in a second. Plus, I explained what is on the histogram. So again, this is the same principle that we learned about editing these strings using annotations and hard-coded text. So now I have a histogram, and now I would like to save this one. Now here you have two options. You can either save this as it is, as an image, or you can convert it into an RGB image, which Ruben showed you yesterday, and save that RGB image. If you have a lot of images, like hundreds, then the latter is faster. So if you convert them to RGB first and then save that as a PNG, that works faster than running it directly. But right now we only have one, so we will take a quick look as a repetition from yesterday of how this one is built. So this is what we do. Basically, I want to automate the saving of this histogram because every time I run it, I don't want to have to worry about it. So I have to create a name for the saved image, and I have to create a target for the saved image. And this is where I find customized folders extremely useful. All I have to do is say, find my analysis folder, which I defined at 9:20 this morning. It was called analysis folder. And the command to call it back, as we saw this before probably already, project data directories. It's an array, and within that I need to find the key to the analysis folder, which we called analysis folder. This is the same name that we set up when we looked at the project overview and we set up the customer folder. And that's all. I do a / to make a subfolder. And within that, I will do a name for the image. And this is up to you 100%. But what I did was I wanted to keep the name of the image so I know where this came from. I wanted to put into the name what I'm plotting there, and this is a hard-coded text median speed. And I also wanted to know what model I used. And the source slot is not the best name, but this contains, for example, in this case, LAP. You saw that there is many models for the tracking. I put it into an annotation which model this was. So the image name will then be generated like that. And once I run this one, this will put the data into analysis folder. Now we have this one saved automatically. It went into the analysis folder. I could have made a subfolder within the analysis folder using the subfolder annotation, but now it's automatically saved. And this is extremely useful when you have a lot of data and you want to then overview what else comes out of the analysis. So basically, from this one, you can go further and process your result table. I do this with the apply expression to columns. The X means there is a newer version of this node, but this simpler version of the workflow I'm showing you now being finished around 3 in the morning, I did not change this one, but it works. And basically what I did here was I processed this output result a bit further. So you remember from the measured tracks, we get this big table with a lot of data. We plotted it, we saved the plot, but we also can do further calculations. For example, we can average the average speed and then get an average value that characterizes the whole population of the neutrophils under this condition, this replicate or this condition. And that's what I do here. I say, take the column, which was called track median speed. This column comes from here. You remember we saw that when I showed you the table. And all I want to do is calculate its average value and save this into the same table under the name of average median speed. So this will be, again, the average speed for the whole video. This could be useful when you want to, let's say, classify results, say, under such conditions, these neutrophils averaged for the whole video moved twice as fast as the other ones. And then the other option is you plot the histograms like we did before, and you compare the peaks of the histograms between conditions. And then I also want to keep this newly calculated value, the average median speed, as an annotation for the video. The video is the image. It has the annotation hash image. I want to add one more annotation, and I want to call it median speed. Why? Because later maybe I will have dozens of videos and I want to limit my further analysis to some with certain median speed. And then I will use this annotation for that. So this is a very easy way to add an annotation. And I also generated another one, which was the standard deviation. So how much does the speed vary between the neutrophils? And then finally, I added a column to the table, which I called condition, and it basically took the median speed and it took the name of the model that I was using. So I'm generating data columns and I'm generating annotations which will help me later to analyze the data and separate them into various groups. So now I have a table which I'm happy with. Okay, so we have the original data plus we have the newly generated table values and annotations. For example, we have the condition, we have the standard deviation, we have the average speed, and so on and so forth. And now I would like to save it automatically. And this is done the same way as Ruben showed you and as I showed you with the image saving, namely, I generate a name the same way as we did before. Same logic, there's nothing new for you anymore. Analysis folder, image name, median speed, and so on. And this will be an Excel file or a CSV file, it's up to you. And when you run it, you will have in the analysis folder a new table. This one, it was just created at 11:25. And it's an Excel table, and it contains, not surprisingly, the values that we just generated. So now you analyze your data, saved your images, saved your Excel sheet. Basically, you are done with characterizing the image. Fairly straightforward, but it really contains every step that you typically use for your analysis. And basically, I was satisfied with this. I added a couple of other options. For example, instead of using a histogram, I generated a line plot. This is the same data as the histogram itself, but instead of using a histogram, I plot the number of cells at a certain speed and make a line XY plot out of it. For this one, we have a node called XY line plot. It works the exact same logic as the histogram. So we don't go through that. And then we save this plot and we save the new table as we did before. Again, the same logic. You can click on your own computer and see what in the parameters. It is exactly the same logic. So now we are very happy. And then we say, okay, this is my output. I will hook this up and then I'm done with the measurements. So where are we? We are done with the measurements. But maybe we would also like to visualize the results. As you notice, I plotted a few histograms and data, but the main visualization I save for a separate compartment. And this is a very simple one. It's still a separate compartment because then I understand what I did better. And if I later decide to add more visualizations, I will add them to this compartment rather than cluttering up my previous compartments. And there is another reason I wanted to show you guys this, how to use ImageJ plugins for which we do not have a node yet, but there is a plugin. And this is to put an ImageJ macro or ImageJ command or plugin call into a JIPipe node. This is not native JIPipe then, but it can solve your problem if you really need something, but it's not available and you don't have a room to develop it for you. Then you put it into ImageJ macro node. So for example, my goal here was that when I have a very long time series, it is possible that the light intensity will change during the experiment. For example, it's a chamber at 37 degrees, humidity 5% CO2. Eventually, you might start to have some condensation on top of the petri dish. The intensity, then the light that goes through the petri dish, will drop over time. So you might end up with an image series where the intensity goes down over time or up. It can also be fluorescence images where you start bleaching the data. The point is that if I want to quantify intensities, I should not have this intensity change over time. And this is called bleaching correction. What it does is it measures the intensity of the image at the beginning of the time series. It measures the intensity at the end of the time series and then linearly interpolates between these two values and corrects the intensity of every time point so that the average intensity is flat. We don't have a node for that, but I needed it. So what I did was I went to ImageJ/Figge and I ran this plugin and recorded what the command looks like. Now, how do you do that? So let's say this is the image that you want to bleach correct. As you see, there is not much intensity change during this, but over a longer time there would be. So I want to figure out how to bleach correct this one. So first of all, what I do is I will put this guy into ImageJ. The way to do it is you open the image in a JIPipe viewer and then you go to export and then you choose your target. In my case, I want to go to ImageJ and I don't have any drawings on the image, so I can just put the raw image. If I had drawings like regions of interest, I could also put the overlaid image into ImageJ, but I go to here. And now what I have is the same image inside Figge. So now I can play with it without having to change my code in JIPipe. I can play with it directly in Figge. And then I say, okay, I want to correct for bleaching. So I do a search in Figge called bleaching. I start typing, and then it says, yeah, there is a bleaching correction command. So I go there, and then I run it. Now, this one will not help me yet because I won't know what command Figge will issue to run this plugin. So what I will do is go to plugins, and I will go to macros, and I turn on the recorder. This will record every command that Figge submits. So I turn this on. This will open a little window for me. This is where the command will go, which I will then copy into JIPipe. Now, I'm happy with this. I go back to my image, and then I go to Figge and I say, okay, I want a command which is about bleaching correction. And it finds it for me. I click on it and I say, okay, run it. There are various methods. I went with the simplest one. So I get the command, how to call this particular plugin. And then what I do is very simply go back to JIPipe, go to my ImageJ macro node, go to parameters, and then I copy this command into here. Just like that. The exact copy of what I recorded with my macro recorder. And this works for all plugins that put out a copyable command. Now, how do you build an ImageJ macro node? When you download or put a new node onto the user interface, it will give you hints. For example, the image that you connected into this node will be called input, and then you have to select that. And then you can put your actual command here, as many as you want. But then you have to put the resulting image into the output slot. And once we have that, it will go through, and it's a macro. So now it's going to flash the images. But at the end of that, we have an image series where the potential bleaching has been eliminated. And as you see, even the flashing that you saw during the video is now gone because this disconnection removed it. And then all I do is, in this particular node, is I just overlay the spots over the image. Very, very simple. The segmentation outline in red. And in the next node, I will overlay the tracks on the original images. And then if you want, for example, you can save these as videos or images, or you can put them to the output. The point is that in a separate compartment now we have the visualization. And then we are done with that. And here, and what I decided to do also was to measure, for example, the distance between neutrophils during the experiment. And I put this into a separate node. It takes the output from the tracking, it has the tracks, and it has the raw images. And basically what it does is it calculates how far the neutrophils are from each other during the experiment. So basically, how dense is the population of these cells, and does it change over time? And that could be a very good measure. For example, cells are dying, or cells are migrating into the field of view, or cells are eating pathogens, and then the number of pathogens will decrease, or their distance will change. So it is a useful measure to see how far these objects are from each other. And one way to do that is to measure the distance between regions of interest. And there is a special node for that, extract raw statistics for pairs. Now, if you want to measure, in this case, the distance between the same population, you need the region of interest, which describe where the objects are, and you connect the same set of region of interest to both inputs, because I want to compare within the population, how far they are from each other. But you could also have two different sets of regions of interest. And you can say, these are macrophages, these are neutrophils, how far are they from each other? And then one row would be the segmented macrophages, one row would be the segmented neutrophils, and you would compare them. Now in this node, this is an extremely useful node because it can measure a whole lot of things. But what we are interested in is the distance between the centers. There are a whole other measures, for example, but this is what we want to do. And then when we run it, then basically we have a data table which contains, for example, cell number 385 from cell number 385 was at 0 distance. That's assuring, it's not surprising. Then the same cell from 375 was at 0.6 micrometers, and so on and so forth. So for the whole movie now I have how far these cells are from each other. And what do I do with that? Well, of course, I will plot it. It's a histogram plot. There is nothing new in it for you anymore. But what you will then see here is the output. And what you see is that, of course, this 0 bar is the ones measuring to itself. So that's natural. But what you will see is that the population in general is between 25 and I would say 55 pixels from each other. And then you can have this either per image or you can have it for the entire video. If we go back to the compartment overview, we completed our project. We have all the outputs, they are automatically saved, we characterized the movement of the cells, we adjusted the tables, we have done everything that needs to be done relatively simply, so I could go through this video in a couple of hours. So this is really showing you how straightforward this can be once you become familiar with JIPipe to develop relatively complex workflows. Now, if you imagine having to do this as an ImageJ macro, I had such a macro. That was one of the first things I did when I moved here. It had 3,000 or 4,000 lines of macro code. And it found macrophages, it found pathogens, it measured the movement, all kinds of things. But can you imagine to maintain and to share 3,000 or 4,000 lines of macro code with people and to teach it at a class? This would not work because even I got lost eventually in that. And that was one of the main reasons that we developed JIPipe, to put all these complex image analysis and data handling steps into a graphical interface that can be much more easily taught and much more easily shared. Because now if I give this to you, you went through it, nobody complained. So this actually worked quite easily. And this I put together last night because I had a much more complex version of it. The point is that within half a day or so, you guys all have it and know how to use it, and you can take it from here and develop it further. You can play with the parameters, you can add additional steps, you can do whatever, because you understood how this works. If I gave you my original macro, you would be like, yeah, thank you, no. We don't want that. So I hope that you learned how to handle larger projects, how to really grasp the power of JIPipe. It is complex, yes, especially with the annotation system. It's not trivial, but once you jump over that hurdle, you should be fine.